Hi, welcome everyone. I'm James Marr, Product Manager for Leica Microsystems, and I'm here today with Marta to discuss her work with the LMD system. Marta, would you like to introduce yourself?
Hello, everyone. I'm Marta Paterlini. I'm a senior scientist in Jonas Frisen's lab, Karolinska Institute in Stockholm. And I joined the lab around 15 years ago. And one of my first duty was to be responsible for an LMD.
Fantastic. So yeah, that's what we're here to talk about today. So could you briefly describe a bit about your research and what you've been doing with the LMD?
So one of the major challenges in developmental biology and regenerative medicine is to understand the origin of cells. And in particular, in our case, we are interested in the origin of new neurons in a human brain, a phenomenon which is called adult human neurogenesis. Adult human neurogenesis occurs in a region, the hippocampus, and requires dividing stem cells differentiated into neural progenitors, and subsequently they migrate and mature into mature neurons. For decades, adult human neurogenesis was supposed to happen only at embryonic level. So it was more neurogenesis, not adult neurogenesis. And it was believed to stop after birth. But growing robust evidence, coming also from our lab in the past, led to the definition of adult neurogenesis. And the presence of adult neurogenesis has in turn raised a lot of questions around identity of neural stem cells and neural progenitors, and the turnover of these cells and so on. And this is when the laser capture, the LMD system, came into place for us.
Great. So can you describe how that LMD facilitated this research that you were working on?
So when we started, it was around when I joined the lab. We wanted to understand deeper neurogenesis, and which meant to go and investigate all these different differentiations of subtypes, leading from a stem cell to a mature neurons in adult samples. And at that time was when single-cell genomics was coming up. We were actually one of the first labs embracing single-cell, the single-cell world, together in collaboration with labs at SciLife, where they were setting up the infrastructures of sequencing. And we reasoned that starting from such a complex system as a human brain wouldn't not been smart. So we were looking for a proof of principle to test our approach, like to define a pipeline of single-cell genomics to define the lineage tracing of human cells. In our case, would have been from stem cells to mature neurons, because it was known that the distribution of somatic variants in single cells can reflect the phylogenetic trajectory. So we started to and we wanted to combine, of course, the single-cell genomic and a bioinformatic tools, develop some bioinformatic tools. to analyze the data. And the work is actually came out as a paper in Genome Biology, where it's an bioinformatic paper where the LMD played an important role, because we found that one of the proof of principle could have been like tracing back the division of a primary fibroblast cell line. So what we did from a very practical point of view was actually very, very cool and long experiment, but very rewarding. We grew cells, fibroblasts, onto the metal frame of the LMD system. We let them attach, grow, and then we recorded with time lapse the division of these cells. And we eventually captured the daughters of all these divisions. We amplified the whole genome. And the Combase, which is the software that has been developed and published, was applied. We know that the whole genome amplification, which was one of the first tools as part of the single-cell genomics approach, very powerful, but gave a lot of backgrounds, like a little dropouts, artifacts. So the idea of this software was to, in an unsupervised way, to discover somatic mutations among single cells. And the LMD was instrumental in building up this pipeline, because we, together with another proof of principle, which were leukocytes from human blood, we tested this software and it worked. So this was the very first work done with LMD, and it led to a publication in Genome Biology in 2019.
What do you find most useful about the Leica LMD for your workflow?
I'm a big fan of the LMD system, and I think it has really the power to work at single cell level. We work at single cell level. And I must say that all the time that we didn't succeed was not because of the LMD, but it was more downstream. Maybe the chemistry of the protocols were not still there. I mean, working with single cells is very difficult, and especially when we move to human brain. Human brain is very difficult to deal with. Even simple immunohistochemistry, immunofluorescence become a struggle. So I think I always say, as I said at the beginning, we own the LMD, but we are generous and we share it with the other people in the department. And along the years, a lot of labs used it. And lately, I feel that there is a new wave of interest in the system. Because in the last 6 months, I was asked by at least 3 labs in the institute to use it, with mainly in mind to test proteomics.
Ah, okay. And then for the omics works, is it mostly mass spec downstream, or is there other things people are doing with those?
I don't know really, but I don't know really what they are, because what I usually do is just to give an introduction, be available if they have problems. So I don't really get into their troubleshooting. But I think, and also by going to seminars, I think the future the present future of the LMD seems to be proteomics applied to mass spec, ultrasensitive. And I think, I mean, now I'm very absorbed by other projects, but I think also for us would be the next step to test proteomics with LMD. I think it could be very important, instrumental in defining like cell function and heterogeneity in health and disease samples, especially with an eye. I mean, there is more and more talking about precision medicine. I think proteomics could be a way to be included in the vision of precision medicine.
That kind of leads into my next question. You've kind of already answered it, but I still want to ask just to see if there's anything else you expand upon is, do you see the LMD in future experiments? You've mentioned proteomics, but do you think it would play an important role, say, in like the spatial omics, spatial proteomics field, things like that? Do you have any insight into that?
I do. I think that in the last 2-4 years, there was a lot of spatial biology, very fancy, and it is. So I have a feeling that because I think you have the feeling, at least this is my personal view, to have hands-on. With spatial biology is very precious, but at some point you risk to draw in data. With the LMD, you have your section, you see it, you laser capture what you see, and then you can sequence it. I think the problem a few years ago was that the chemistry downstream, especially for single cell, was not sensitive enough for one tiny cell sometimes. So, but overall, I think LMD has a strong space still in spatial biology, and definitely in new lines like proteomics.